So all these students that you're going to hear from today all did summer research, and I think you'll learn a lot about their experience, and hopefully you can take a little bit away from what they did. And if you're interested in doing summer research, please talk to the students afterwards and come talk to the faculty as well. So we'll start off with Catherine Craig. Give it a second. It's coming. The suspense. No signal. Is that better?
So, like Dr. Walter said, I'm Catherine, and I did summer research with Dr. Pirtle, who's sitting over there, through the INBRE program. And the title of my project is kind of long, but it's Exposing Daphnia Magma to Ethanol Results in a Decline in Heart Rate by Increasing Nitric Oxide Production. So, the organisms I worked with this summer were Daphnia Magma. That's a very distinct picture of them. They're actually really small. They're about 1 to 2 millimeters in size. So we had to use very strong microscopes to be able to see them. They are crustaceans, but even though they are crustaceans, their hearts are actually very similar to vertebrates in that they're myogenic. So you can have two kinds of hearts. There's myogenic and neurogenic. And neurogenic means that the heart is getting a stimulus from the nervous system to contract. Whereas myogenic means that the pacemaker is within the heart, which is very similar to vertebrates such as us, that our heart beats on its own without getting the nervous system stimulus. So because their hearts are very similar to vertebrates, it makes them great model organisms to work with when wanting to test different chemicals on the heart and see what it does to them. So this is kind of a very complex diagram of what the Daphnia look like. The big things, let's see if this works. So this is their heart right here. It's kind of like where the back of our spine is, where our neck is. And I'll have a picture to show you what it looks like underneath the microscope. And then this right here is called the Carapace, which is this clear casing around them. It's their exoskeleton, and that is what we used to pin them down without hitting any of their major organs and potentially harming them more than we are. So, like I said, we can use several different chemicals to test their effects on the heart. This is a project that's been going on for several years with Dr. Pirtle. So this summer we wanted to look at nitric oxide. And nitric oxide is a chemical signal that's found throughout your entire body. It's used in a lot of different processes in your body. But like most chemical signals, and I'm sure most of you guys have talked about this in class, if it's in excess, it can cause harm to your body. So nitric oxide has been linked to a lot of neurological disorders like Parkinson's disease and multiple sclerosis. The belief is that the excess nitric oxide leads to increased free radicals in your body, which can affect the cellular membrane of your cells. But while it's been studied a lot in relation to the brain, as far as we know, it hasn't been studied a lot in relation to the heart, which is why we decided to look at it this summer. So our hypothesis for this summer was that exposing our Daphnia to a nitric oxide donor would increase their heart rate, and then exposing them to nitric oxide blockers would decrease their heart rate. And this is kind of a schematic of our methods. So like I said earlier, we pinned the Daphnia down into petri dishes using cactus spines, and we pinned them in their carapace so we wouldn't harm them. And then we exposed them to 4 different chemicals using a perfusion system. And those were a spring water control, our nitric oxide donor, nitric oxide blockers in ethanol, and then a 10% ethanol control. When we started the project, we thought one of our blockers had to be dissolved in ethanol to go into solution, which is why we had the 10% ethanol control to go with it. Otherwise, we wouldn't have needed it. So once we exposed them to these chemicals, we took 5 15-second videos of their hearts and stored them for later use so we could count their heart rates. And when we got all of our data collected, we then sat down and slowed down the videos and timed or counted their hearts manually using those little hand timer things. So this is what they look like underneath the microscope, and it's going to be kind of hard to see, but you can see their heart is right here, and this is one of the videos we took. And so this is what we would sit down, and we would slow it down and just hit the button every time that their heart contracted. And this black spot right here and right here are the cactus spines that are pinning them down so they're not moving in the middle of our video. So that was a 15-second video. We did 5 of those for each of our Daphnia. We did 10 Daphnia for each chemical, so there was a lot of sitting down and counting heart rates for the summer, which was great. So for our results, this first graph is depicting what the nitric oxide did to their heart rates. The orange line is their spring water control, and you can see that it didn't really change very much. Their heart rate did decrease a little bit, but nothing substantial to cause any worry. But their nitric oxide donor decreased significantly. After the 20 minutes, we found extreme significance, meaning that it was very different between the ethanol or the water control and the nitric oxide donor. And then this graph is depicting our nitric oxide blockers and our ethanol controls. So the orange line is our spring water again, and then the blue line is our nitric oxide blockers with ethanol. And you can see that it decreased as well, which shouldn't have happened. The theory was that the nitric oxide blockers would either increase their heart rate or keep their heart rate steady because it was preventing nitric oxide from being produced. But if you look, that our nitric oxide blockers is this kind of pale blue line, their heart rates decreased immensely over the time of 20 minutes. This wasn't really supposed to happen. We weren't supposed to get any data from our ethanol. It was just meant to be a control, and we ended up getting the best data from our ethanol. So goes to show you never know what you're going to get when you do research. And so because our ethanol decreased so much and our nitric oxide blockers decreased as well, our theory is that, or our hypothesis is that the nitric oxide blockers did work to some extent, but they weren't able to work to their full extent because they were present in ethanol, which mitigated their process. So our big conclusions for the summer was that our hypothesis was very wrong, and that nitric oxide decreased their heart rate. We're not sure why. That's something we're going to keep looking into to see what about nitric oxide is leading to their heart rates decreasing. We also found that ethanol decreased their heart rate a lot. So we're going to keep looking into that. And that the exposing our Daphnia to our nitric oxide blockers did decrease their heart rate, but not as much. And we think that they weren't able to work because of the ethanol being present. So from all of these conclusions and our data, we were kind of able to make a broad working hypothesis that ethanol actually increases nitric oxide production because it stimulates nitric oxide synthase. And we're not sure how. There's kind of a little description right there. I won't go into it. But so that's kind of something we want to look into further. So what we've done since this summer is we ran the nitric oxide blockers again without ethanol. Once we did all of our experiments and did all of our data and stuff, We looked closer at our chemical and found that it didn't need to be dissolved in ethanol. It can be dissolved in water. So we did that with not needing to, basically. And so we've redone all of those experiments, but I haven't gone through the data yet, so that's why it's not included in this presentation. We also have looked at the effects of cyclic AMP and cyclic GMP as well, and that data is somewhere too that hasn't been gone through yet either. But basically the whole premise of what we're wanting to do in the future is follow the nitric oxide pathway to get a better understanding of what about that pathway is leading to their heart rates decreasing. And then there's also an assay we found that can calculate how much nitric oxide is being produced, so that might be something we try in the future as well. Those are my references, and thanks Dr. Daniels for letting us use your lab. And, yep. Are there any questions?
Why did you use cactus things to get involved?
Because their cactus spines are really small, and they can penetrate their exoskeleton quite easily without harming them, as opposed to using metal pins that are fairly large. And because they are so small, we needed something that was the same size, basically.
So I'm curious, what is Daphnia? Where'd you get them from? Saltwater?
They are freshwater crustaceans. Yes. We didn't get them ourselves. We ordered them online from Carolina Biological. So, but that's where you can get them.
So when you dissolved your blocker in ethanol, you used 10% ethanol, right? And was your control also 10% ethanol, or was it just straight ethanol?
It was 10% ethanol. We tried to keep everything consistent, so we weren't having any big ranges.
I got a quick question.
Yep.
So why did you think that nitrous oxide would decrease heart rate and not increase heart rate? So you made a pretty specific prediction.
Mm-hmm.
And I'm just wondering what you based that on.
So we hypothesized that it would increase their heart rate.
Okay.
And there was a paper that was written a long time ago that it was looking at the effects of denervated hearts, which are hearts that are being transplanted, and they tested nitric oxide on those, and it actually increased their heart rate. And because denervated hearts are very similar to the myogenic hearts because they're getting their stimulus from inside, that was why we hypothesized it would be increasing as well.
Okay.
And Dr. Pirtle in the past has proven that these Daphnia are a good model organism because they do have very similar mechanisms in their heart. He's got a paper that's upstairs about it. So that was why we hypothesized that.
Okay.
Yeah, so I want to know, what were you trying to do the heart rates? What's the implications for us? What's the, I don't know, what's the, how does it relate to us?
Right.
How does it relate to
How
us?
does it relate to us?
Yeah.
So this is very new. We're starting from scale one type thing. So this would be a long time before it ever reached to anything having to relate to us. But in understanding the nitric oxide pathway, we could understand nitric oxide pathways in our bodies. There's been, I was talking to Dr. Pirtle about this morning. There's been some research trying to look at the effects of nitric oxide in diabetes, and that they have found that people with diabetes tend to have greater concentrations of nitric oxide. So if we could understand what about that nitric oxide, what the nitric oxide pathway, what it's being stimulated, we could hopefully figure something to do with diabetes.
All right. Thanks, Catherine. who's next? You snuck in there. I didn't see you.
Hi, I'm Kaden, and I did research over the summer about the effect of MFD on 3 paraquat tolerance genes during disulfide stress in Bacillus subtilis. MFD is a gene that codes for the DNA repair factor in Bacillus, and then I was looking at 3 genes in paraquat tolerance, which is a oxidant, and disulfide is also an oxidant. So I'm just going to go over like what it was and then my project. So I was able to do this through a research education opportunity for undergraduates, RU, called Mechanisms of Evolution, hosted at UNLV, the University of Nevada, Las Vegas, and funded by National Science Foundation. And they basically are a branch of the federal government. They give grants to institutions to fund undergraduate research opportunities, and they give these institutions a bunch of different requirements. So we participated in a book club on Jerry Coyne's Why Evolution is True, and then we also did like lectures about, or watched lectures about grad school, and in addition to the projects. So my project was on this specific gene MFD, which stands for mutation frequency decline. And there's a picture on that side about how it works. Basically, during DNA transcription, if the RNA polymerase that's transcribing gets stuck on a DNA lesion, then it can either push it over that blockage, or it can knock off the polymerase and then let other things come take care of that problem before it reattaches. And that is a DNA repair mechanism. And one of the other members of the lab, Holly Martin, they just published a paper in January about this gene that codes for this DNA repair factor impacting tolerance against oxidative stress. And they wanted to explore that more because the known function of MFD was only associated with DNA repair. And larger impacts, NSF was funding this program, so looking at like kind of general basic science, like just learning more about the world around us, in oxidation, which occurs in all organisms while we're alive because it happens as a part of respiration. And then I was working in Bacillus, which is a model organism, and the focus of the projects were in evolution, and MFD is involved in evolution because it's involved with the DNA repair. So my hypothesis was that MFD has an epistatic effect on ALD-Y, CYP-C, and YFHE, which were my 3 genes, during oxidative stress. And epistasis is when one gene impacts whether or not another gene is like active. So the like really common example is the puppies, and there's one gene that controls whether or not pigment is added to their fur. So if that gene is recessive, which is no color, then the puppy is going to be yellow whether or not the black or brown gene is turned on. And then if the color gene is turned on, then the black or brown color becomes relevant. So how I addressed my hypothesis was by creating a whole bunch of bacteria mutants and then looking at different gene deficiencies. One set which were lacking the gene I was interested in, and there were 3 of those, and then one set missing MFD and also missing the gene I was interested in. So these were my genes. I'm not going to go through all this stuff because there's a lot of numbers that don't really mean a whole lot, but the name of the genes are up there, ALD-Y, CYP-C, and YFHE. And one of the interesting things was that YFHE is like the teensiest little gene ever. It's 108 base pairs. So in like the picture, the genes are labeled, and it's circled in red because it's so small it doesn't even have its name written on the gene number. And my methods on how I tested this. So we ordered existing bacteria strains that were already mutated to be missing the gene I was interested in, and then pulled that out in the form of genomic DNA and transformed into our lab wild type, which was like our regular bacteria. And we did that just to make sure that everything's consistent, so we're not working with a mutant in one bacteria type and then comparing it to another bacteria type that aren't the same. And then the other combinations, we have an existing strain that's missing MFD, and then creating the mixed one, where we took the gene deficient DNA and moved it into bacteria that were missing MFD. So we end up with 4 regular wild type minus MFD, and then minus the gene, and MFD also minus gene. And we took all of those and exposed them to diamide, which is an oxidant. So this is just how transformation works. We put DNA in the environment of the bacteria so that it can be taken in and integrated into the genome so that that bacteria now has the traits of that DNA. And then the graph on that side is the way that bacteria grows, because we tested during stationary phase, which is the flat bit on top. So bacteria, when they're in their media, grow and continue growing until they start running out of resources. And then basically the bacteria apocalypse happens, and they start doing crazy things like eating each other and uptaking DNA from their environment, which is how we were able to transform them. And after that, they start running out of food and start dying off. So these were my results. The letters are for statistical significance. The letters basically mean that only wild type was different from anything else. My expected results are up in the corner. We thought that we'd get a really tall bar for wild type, a really, really tiny bar for MFD minus, and then the single genes would have something in the middle, and the single gene also without MFD would be super, super tiny like MFD. And my results came out kind of wonky. My MFD and most of the ones that were missing MFD were very similar. And then the bars appeared to be different, but were not statistically significantly different. So based on that, we decided that they do contribute to stress tolerance, because when we took out those genes, they didn't survive as well in their environment. That CYP-C, which was the tiniest bar other than MFD minus, is the gene that contributes most to tolerance. And that MFD might have an effect, but the numbers weren't significant enough to draw that conclusion yet. And if I had more time, which I really wish I had more time, I would have liked to do a lot more trials and gotten more data so I could have statistically significant results. I also would have liked to test different oxidants other than diamide, like paraquat, which is what these genes are associated with and probably should have been testing with. But all of the other data that we had in the lab were based on diamide. And then I also would have liked to find out if these 3 genes have any impact on DNA repair, because we selected them based on being associated with a DNA repair factor, which was kind of odd because they are known to be involved with protein protection and not DNA. I lost everybody, and it was really confusing. So I would like to thank the Robledo Lab and UNLV Life Sciences, Dr. Kurt Regner, who is the program director, NSF, NIH, who funded, and I have the grant numbers at the bottom. So, questions?
Can you elaborate a little bit on what oxidative stress is for some people here?
Yeah, so we used a chemical called diamide to test whether or not the cells would survive. And diamide causes a chemical reaction that damages the protein in the cells, or in the bacteria, I guess. So it damages it, and then it can't survive as well. What diamide specifically does is it starts adding thiol, like sulfur groups, to everything. It just starts adding things onto it, which breaks the proteins. They don't function properly. And we were able to measure how well they survived by counting the number of bacteria that survive. applied for like 8, and I got 1 acceptance letter. But I wanted to do something related to microbiology because that's something that I'm wondering if I'm interested in. So that was, yeah, and there's just a lot of these programs, so just the applications are individual.
Did it make you more or less interested in doing that then afterwards, the microbiology?
I don't know. Okay, I really liked working, and it was really fun. The lab environment was kind of weird because everybody in the lab went on vacation at the same time, right when I got there. So I was in a lab that I'd never been in by myself for about 2.5 weeks. There was one girl who showed up 3 times to show me how to do things, and she was like, yeah, this is how you do stuff. Okay, you're good, right? Here's my phone number, bye. And that was it. So I was just kind of running this assay over and over and doing transformations over and over, not sure that I was doing them right, just kind of kept going. And then they were like, oh, wow, your numbers are fine. And I was like, okay. And that was like halfway through the program when everyone got back from vacation. So, yeah, but I found it really interesting, and a lot of the stuff, information, like, just totally went over my head. So then they started asking me questions like, what is disulfide actually doing? And I was like, I don't know, you gave me a bottle called disulfide and told me to add it, so I added it. And then my PI would pull out a textbook and throw it at me, so, yeah.
Any other questions? All right.
So I'm Andrew Wymore. I worked with Dr. Walzer this summer studying the effects of flow cessation on red band trout. It was a really great opportunity. I was really excited to get the opportunity. I have a pretty good relationship with Dr. Walzer, and that got even better this summer. And this is kind of what I want to do for the rest of my life. So yeah, really great opportunity for me.
Hi, my name is Lydia Brown. I'm a junior and I'm a bio major. And like Andrew said, we got to work alongside Dr. Walzer. And one other student researcher who couldn't be here this evening. And we were studying the effects of flow cessation on red band trout in Dry Creek. So in terms of our research question, our formal research question that we chose to investigate was that we were investigating the response of Columbia River red band trout, so the trout that are up at Bogus, and macroinvertebrates to the changing physicochemical conditions in Dry Creek, Idaho. So basically in some, as the summer goes on, the flows begin to change. As heat increases, that means that there's going to be less water in the stream. So then in terms of our study importance, one of the first reasons that I think it's pretty easy to understand the importance of our study is we're kind of examining the effects of iris system ecology. What is an iris system? So as we define up here, iris is an intermittent river and ephemeral stream. So basically what that means is that it's a waterway that doesn't have flowing water all year. So what's really fascinating is that like most perennial waterways, or waterways that have water flowing all year, like the Boise River or most larger waterways that most of us can think about, that's what most like research about waterways is about, and there's actually surprising lack of studies on iris systems, despite the fact that 1, they're increasing with climate change, but 2, they already are the majority of water systems in the world.
Yeah, and then the other implication that Lydia mentioned is conservation. So climate change, we all know, is a big problem, and as the climate changes, that affects how waterways function and how the flow actually moves through the waterway. So as climate change happens, it's getting hotter, the flows cessate more, and there's less water flow going through Dry Creek, which increases the stress on aquatic populations and the macroinvertebrates and the fish. So basically, here's a kind of schematic of what happens when temperatures increase. As air temperatures increase, the riparian vegetation here, so basically trees, bushes, grasses, they intake more moisture from the soil, which decreases the amount of water in the stream, and then it's evaporated into the air as water vapor. So basically, that's how the streams lose a lot of the water as the summer progresses and temperatures increase.
So this is a map of our study area. So basically just to kind of give you an idea of how we chose to actually investigate our research question. So Dry Creek is a tributary to the Boise River, so it's kind of flowing down Bogus Basin. Most of us, I think, have probably gone up to Bogus for skiing or whatnot. So Dry Creek goes down there, and so we chose 10 study sites along Dry Creek, and the way that we chose these 10 study sites is that they were basically 10 separate locations along the creek in which we hypothesized at the beginning of the summer that those 10 separate locations were likely to become individual cessated pools as the flows began to decrease, right? Because whenever the summer goes on, flows begin to decrease, and whenever that happens, there's likely to become more poolings of what used to be the flowing river or stream. And so most of our sites are kind of in between. It's like this nice little 3-color areas where our sites are. And so most of our sites were within, or our sites spanned within like 5 kilometers. And so each individual site, like site 1, site 2, site 3, and so on, were between like half a mile to just a couple hundred feet from each other.
So this shows just kind of a comparison. This is from a few years ago actually, but this picture was taken in June and this picture taken in August. So as you can see, there's a lot of drying that occurs because obviously this had water and then 2 months later it doesn't, and the flow is completely stopped. So these conditions are really stressful for the fish because there's less oxygen in the water and it's easier for the water to get hotter as air temperature increases.
It's hard to be an aquatic animal when there's no aquatic.
So for our methods, for our fish surveys, we use an electrofisher because it's kind of the most efficient way to catch fish. Basically, it just shocks the fish and stuns them to where we can collect them with a net. And then after we collect them, we anesthetize them if they need, or anesthetize them to calm them down so we can weigh and measure them. And then we also tag the ones that were big enough to tag to allow us to track their growth and survival over the summer. And then we also, for our macroinvertebrates, which are the bugs that the fish eat, we use drift net sampling as well as server sampling. Drift net sampling allows us to kind of get an idea of how many insects are actually flowing in the water, and that's what mainly the fish are eating. But the server sampler also gives us an idea of how many bugs are kind of in the sediment of the stream bed because we disturb it as we're sampling, and they kind of like come up out of the sand and sediment. And then we took the macroinvertebrates back to the lab and identified them using taxonomic keys down to either the order or family level.
So in order to investigate basically what was happening to the organisms within Dry Creek, we had to take physicochemistry data. So this slide is kind of nice because it divides up our physical habitat characteristics that we were studying as well as our water chemistry characteristics. So basically what was happening is, like I said, we had 10 different site locations. At these 10 different locations, we revisited them multiple times throughout the summer and collected these different data points. So things like length was kind of like pretty constant, the length was pretty similar throughout the summer, but things like width, depth, and current velocity substantially changed throughout the summer. So those are things that we collected in order to monitor how the pools or the stream was actually changing. And that's also true with the water chemistry data. So for water chemistry, we collected temperature, dissolved oxygen, turbidity, conductivity, pH, total ammonia. And we used all these things with handheld devices. But additionally, partway through the summer, we were able to get these loggers that we placed permanently in the stream. And so they were placed in the stream, and they were able to collect temperature and dissolved oxygen data every 30 minutes throughout the summer. So we were able to basically create a pretty comprehensive data set monitoring the temperature and dissolved oxygen levels within the stream, even when we weren't able to go up to Dry Creek and collect that data. So for our first result, I think this is a pretty simple way to kind of look at a basic change that was happening in Dry Creek. So it's basically the change of current velocity at one of our sites. So like I said, we had 10 sites, so this was all measured at our first site, site 1, and this is all monitoring how current velocity changed throughout the summer. So as you can see, at the beginning of the summer in June, the current velocity was at its peak. That was the highest current velocity that we found throughout the whole summer, and then obviously it comes all the way down to 0 towards the end of August. So that really shows you how like the velocity of water substantially changes as heat increases throughout the summer. And this, we had very similar trends at the rest of our sites, obviously, as they all experienced the same summer. And so this is one of our other graphs that we have. So this is looking at dissolved oxygen and temperature variation across time. This is also at Site 1. So like I said, we had loggers that we placed at certain sites that were able to collect data points every 30 minutes. And so that's why this graph has so many data points, because they were collected every 30 minutes. So the blue line is the temperature, and the black line is dissolved oxygen. And so temperature and dissolved oxygen have a relationship with each other. So firstly, warmer water has less dissolved oxygen. So in the first place, the summer is already hard on aquatic organisms because the warmer the water is, there's less oxygen available in the water. But secondly, whenever temperature increases, it also affects the flow. And whenever there's a decrease in flow, there's less of an interaction between the water and the air. And whenever there's an interaction with the water and the air, the water is able to get oxygen from the air. So whenever flows decrease, there's less oxygen being put into the stream. So that's another stressor on how there's going to be like less oxygen within the stream. And that's demonstrated here perfectly. So as you can see, like I said, the blue line is temperature. So you see that spike in temperature right up there. That was kind of like those hot days right when class started and we all thought it was really hot. That was like what was happening right there. So temperature was really increasing, and then obviously there was a massive decrease in dissolved oxygen. And that is a massive stressor for the fish because obviously there's not a lot of oxygen available for them in order to breathe in the water. And then as temperatures began to decrease again, so you see this decrease in temperature right here, obviously the dissolved oxygen went back up. So basically what happened there is that flows were able to go back up because it wasn't as hot. And so with that, whenever those flows increased, the water was able to interact with the air again and allow there to be more dissolved oxygen present in the water, allowing the fish to breathe.
So this image right here is one of our macroinvertebrates. We got a lot of these guys over the summer. They're Heptagenidae, they're part of the mayfly order. And then this graph over here shows kind of the general trend in macroinvertebrate density in the drift. So what's flowing through the water, basically the food supply that the fish have over the summer. We're not really sure why we saw this dip right here in July. That's 7/18, 7/25. We're not really sure why we saw that dip, but the overall trend is downward. So basically as the summer progresses and as temperatures increase and flows cessate, there's less food for the fish, adding on to the stress that Lydia mentioned of dissolved oxygen and increased temperature. And then this is kind of a summation of that stress. So fish condition is a general measurement of fish health based on length and weight. So as you can see in July, earlier in the summer before it got really hot, fish condition was higher, and then as the summer kind of continued, fish condition on average gets lower. So that's kind of what we were expecting to see.
So in terms of our future goals, unlike some of the Embry folk that have presented, we haven't had the opportunity to present our data yet at a conference that's coming up in November. And so we're still kind of collecting our data and constructing our results in order to present it at that conference. So with that being said, for me at that conference, I'm hoping to investigate the response of feeding traits of macroinvertebrates to flow cessation. So basically what that means is obviously, like I said, whenever there's like a less aquatic habitat, there's going to be less aquatic organisms. So we're probably going to see an increase in terrestrial macroinvertebrates versus aquatic macroinvertebrates. But not only that, but the macroinvertebrates that are going to thrive whenever flows decrease are going to be because of maybe the way that they consume their food, right? So I'm kind of looking at investigating the ways in which these feeding groups are similar and the ones that are favored whenever there's less flows happening in Dry Creek.
Yeah, so like Lydia said, we're planning on going to the Murdoch Conference in November, and then the Western Division of the American Fisheries Society in April of next year.
It's our little buddy we made in Dry Creek. Andrew's buddy.
Oh, and then we'd like to thank the Murdoch Charitable Trust Foundation and the Idaho Gem 3 National Science Foundation Fund for funding our research this summer and our equipment and stuff like that. We'd also like to thank the College of Idaho and Dr. Walzer for being our instructor this summer.
Providing us with a lot of knowledge and
Yeah.
guidance.
Are there any questions? Yeah.
How many fish did you tag?
Tag? I don't know an exact number off the top of my head. Do you have a guess?
No, I mean, like at some sites, you know, if they were like smaller, I think it was like 110 centimeters or something was like the cutoff. So if they were really small, we didn't tag them. And then towards the end of the summer, we also didn't tag them. But I'd say like, you know, at some sites we'd catch like 7 fish, which was like a lot. Then, yeah, and you know, multiply that by 10. There's like 10 sites, and then you know we get 4 or something at other sites.
So you said you're still tracking them today, like.
Yeah, I mean we can.
Okay, because I was gonna ask if you knew like the survival rate of the fish you tagged.
There was, well, Dr. Walser has tagged fish in Dry Creek before, and I think there was like, Dr. Walser said it's really rare to get a fish that's been tagged like multiple years back, and so I don't think the survival rate's much far past like 5 or 6 years.
Yeah.
Yeah.
Can you expand upon how like the deoxygenation of the water works? Is it just like a natural cycle that occurs?
Yeah, so basically warmer water has less affinity to hold oxygen, and also as flows cessate, the aeration that occurs, like, you know, like white water rapids, like there's a lot of diffusion of oxygen into the water. So as that stops and as like water levels kind of like calm down, there's less of that oxygen from the air being mixed into the water. So yeah, it's basically just a natural cycle that occurs. Yeah.
I have a question related to that. So you had a kind of like a graph of oxygen, dissolved oxygen over time. Yeah, is every oscillation like one day?
Yeah, so there's like Deal oscillation, because obviously as like, there's like the natural relationship between temperature and dissolved oxygen, and so like as temperatures like get colder at night, dissolved oxygen goes up, so that's why it's like so like jagged up and down, because it's recording every 30 minutes, but yeah.
I don't know if this is true, but just looking at this, I'm pretty sure every one of these little lines is like a day.
I think so. Yeah, I think so.
Yeah.
Related to that, have you thought of plotting like one time every day over time sort of thing?
Yeah, so actually this was pulled straight from the logger, so we didn't even make this. So like before we made this, like so going back to like the methods section, like I said, so this logger collected data every 30 minutes, but we also had the handheld devices that we used to collect dissolved oxygen and temperature. And we basically just kind of did that to like kind of have 2 point references for dissolved oxygen and temperature. But we have all these data points, so we, you know, in the future we could isolate specific days, or, you know, that we want for the data, but we kind of just have this compilation of data that we're still trying to, like I said, we haven't presented it yet, so I think we're still trying to figure out, you know, okay, do we want to do it by day or by hour, or, you know.
Yeah.
So most of the rivers here are like fed by snow melt and that kind of stuff, and that occurs with rising temperatures. So how does that affect the amount, since the increase of water would help the oxygen?
Right. So I'm not sure really, would you mind like rewording that?
Okay, so
Sorry.
you know how most of the rivers here like are kind of dependent on snow melt from the mountains to feed water into them? You said that since the rivers kind of dry up as it gets warmer.
Right.
But the warming also causes snow to melt.
Yeah.
So how does that affect?
Right. Well, so I think, you know, I'm just kind of going to go ahead and guess on this, but like, I know that, you know, Bogus Basin, for example, you know, even whenever we hiked up there at the end of May, towards the top of Bogus, you know, there's not a whole lot of snow up there, even at the end of May. So with that being said, I think that the rising temperatures initially help increase flow, right? That's why we see a large amount of, you know, that's why we see the Boise River really like flowing a lot at the beginning of the summer, you know, because there's a lot of snowmelt happening. But relatively after that, like in June and July, and especially August, when the temperatures are really high, all that snowmelt is gone, so all that's happening then is like how Andrew talked about, is the trees need water and the riparian vegetation need water, so they start sucking that water from the below the ground by the stream, you know, through evapotranspiration. So ultimately it does end up like, even though the warming does provide water initially, it does end up taking it away.
Yeah, so like the snowpack melt and like spring precipitation, that kind of adds to like the positive force of like how much water is going through initially, like Lydia said, but then like just kind of compounding upon what you said, as the summer progresses and it gets hotter, the kind of negative force of, you know, evapotranspiration, as well as like well water pumping, can kind of pull water out of the stream, so they're just kind of balancing between the two, I guess.
All right, we need to move on. Thanks, guys.
Alright, hi everyone. My name is Shania, and I am a junior here. And for the last 2 years, I've been working in Dr. Daniel's lab studying glioblastoma. And I've also been able to do 2 summers of IMBRI research, which is the same as Catherine's research. So we've been funded by IMBRI these past 2 years. So we've been studying glioblastoma, and glioblastoma is a type of primary brain tumor. So primary brain tumor means that the brain tumor originated in the brain. So a lot of times, brain tumors start out in other parts of the body, and those cells travel to the brain and form brain tumors. But this is a little different because it originates in the brain. It's also the most common and deadliest form of primary brain cancer, and there are about 12,000 new cases each year. And currently, treatment includes chemotherapy, radiation, and surgical resection, but even with all these treatments, most patients won't survive more than 15 months past the initial diagnosis. And because of the reason why it's such a low survival rate is because the recurrence of the tumor is very likely. And there are 3 reasons why the tumor may come back. The first is there's rapid proliferation of the cells, and that means the cells in the tumor are constantly dividing and growing so quickly that oftentimes it's really too late to treat it by the time it's caught. And the second is it has strong metastatic capabilities. And what that means is the tumors themselves are surrounded by a layer of cells that break off from the main tumor and travel to healthy brain tissue and form a new tumor there. And probably the most important cause of recurrence is resistance to chemotherapy. So almost 100% of people that die from glioblastoma is because their tumor becomes resistant to the chemotherapeutic drug, which is called TMZ. That's because the cells that survive the initial dose of chemo are resistant to the chemo, and by natural selection, they're going to continue growing until ultimately you have a tumor that can no longer be treated by chemotherapy. So how is glioblastoma studied? Well, there are lots of ways to study brain cancer. Sometimes you could get data from human patients. You could study a model in a mouse, so you could find the brain tumor in a mouse and study the mice there. In our lab, we study it with cell culture. So it's a glioblastoma model system, and what that means is the tumor in 1970 was found in a mouse. And the cells in that tumor were extracted, and over time these cells have been divided to keep this cell line going. And they go to different labs so that labs can do testing on them and run different experiments to see how they might be characterized. And with this, we could treat these cells that we grow in like petri dishes, we could treat them with chemotherapy and other drugs. And the ideal goal is that it will change the phenotype, and we could try to see how they're responding to these drugs to hopefully characterize the tumor a little bit better. So as I said before, resistance to chemotherapy is a really huge problem, and many patients die because of this resistance. So how can cells become resistant? So what are ways that they'll become resistant to chemotherapy? The first is they can physically pump the drug out of the cell. So if they can pump the drug out of the cell, it means that it's really not going to affect any of the cellular processes, and it won't try to kill the cell at all. The other is that most chemotherapeutic agents, including TMZ, they act by causing DNA damage. So that's going to affect the way proteins are synthesized, and that's going to affect the overall health of the cell and hopefully kill it. And the other is the damage itself won't occur. So you can either not have the drug in the cell at all, you can fix the damage it causes, or not cause the damage. So it's important to know how this resistance occurs, because once you can characterize how the resistance occurs in these glioblastoma cells, you can hopefully find stronger targets for treatment of glioblastoma and hopefully combat resistance. So my project has been trying to characterize and combat the chemo response and the resistance in these cells, in our cells that, again, like I said earlier, are derived from mice. So my hypothesis was first that TMZ will decrease the viability of the cells, and I wanted to find that by using cell counts. So the way I did this was I took a flask of cells that I've been growing up, and I counted how many were there. And then I would add the treatment, and after 24 hours I'd count them again. You know, in theory most cells would die off, and so the number would go down. However, you can see that the results are not very reliable because the numbers are all over the place. We have a huge error, and the likely cause of this is I couldn't see any changes in the cell viability with increasing doses of the drug. And that's because so many cells were dying that any change was really like a huge portion of whatever cells were left. So the next step was to find another way to find how the drug or TMZ affects these cells. So I decided to use an MTT assay. And an MTT assay is a really great way of measuring cell viability and how whatever you're treating the cells with are affecting the amount of cells that survive. So what I did is you could see in the picture to your right, that's an MTT assay, and each one of those wells has the same number of our glioblastoma cells in them. And the thought is, once you grow them all to the same number of cells, and you treat them all with different levels of chemotherapy, you're going to add a dye called MTT. MTT will change to a purple color when it reacts with metabolic products of a cell. So the more drugs you give it, the more cells are going to die. Ideally, that's our hypothesis. So you can see the super purple wells towards the right. That means they have a really high absorbance, which is what our quantifiable value is. So at a really high absorbance, that means you have a ton of metabolic product, and that means not a lot of cells are dying. So we can run these through a meter, and it'll show you, a high absorbance, high survival rate. And in fact, this did help us give clear data. You can see on this graph, we have a decrease in our cell viability, so that's our percentage of cells that survived, with an increasing dose of the drug. And at about a 1 millimolar concentration of the drug, about half of our cells are surviving. So why, this seems like something obvious. Yeah, you're going to treat, a cell line that's not resistant to drugs with chemotherapy, and they're going to die, and that's pretty obvious. But we needed this information in order to go to our next step, which would be to take these cells that, you could see they are being affected significantly by this drug. We want to take these cells and gradually increase the dosage of chemotherapy they're exposed to make them resistant. So our hypothesis would be that if we were to create some resistant cell lines, they'll show changes in their DNA expression, and we could hopefully try to isolate certain ways that our cells are resistant to chemotherapy. So the way we plan on doing this is, you can see if this were a normal flask of cells, we gradually increase the dose of chemotherapy in 2 separate cell lines to see if they'll change their expression independently, and we'll also have one as a control group that doesn't change at all. And the thought is the 2 red cell flasks there will have changes in their DNA because by natural selection, in order to survive the chemotherapy, the ones that are left behind are naturally going to have different mutations that are allowing them to survive. And that will eventually translate into different protein expression. Fun story, we've tried this for about a year now. And we've tried to make cell lines that are resistant to chemotherapy. It has not worked because it did take a year to figure this out, but we made 2 cell lines that were resistant to the stuff that we're dissolving the drug in. So that's not very useful because we're not really looking at how they're resistant to the drug, we're looking more at how they're resistant to what we use to dissolve it in. So now we're starting from scratch, and with the data that I showed before, we now know what concentration we can start at to hopefully combat that issue. So there are some things that I've learned and loved about research. One thing that was surprising to me is it's really different from class labs. It's instead of having a new project every week, you have one project that you're working on for a semester or multiple semesters that you really become invested in. And because of that, it really helps with gaining a deeper understanding of the material. So it's such a specific topic that I've been researching that it's really fun to know a lot about it and try to expand your knowledge on it and use that knowledge to create new experiments and new projects. It also points you in career directions. I never thought I would enjoy research, but I started when I was a freshman, and it really made me realize how much I enjoyed that field of biomedical research. And because of that, now I know it's something that I really want to incorporate in my career in some way, shape, or form. And it's also something that, it's totally doable with sports and everything, so it's nice to have something to do with an academic focus that you could still be passionate about. And on top of that, you meet great people, you meet great professors like Dr. Daniels and awesome lab partners, so highly recommend. But any questions?
Are there any possible differences between using a mouse cell Yeah, so great question. So the human cell line will come with a lot more regulations in terms of like safety and ethics and all that. The mouse cell line, it'll give a good model as to how glioblastoma might behave in a human, but there are a lot of steps between finding something about this cell line versus finding it in a human cell line. So the next step, if there was some kind of significant change in protein expression, would be to see how that affects a mouse when the tumor is actually in the mouse. And from there, you could probably you could probably see how it would affect the human cell line, because phenotypically and genotypically, they're all going to be different. So it's something that you'd have to explore separately.
How did you determine that the cell lines were being resistant to the solvent and not the TMZ?
So what we did was we did a control experiment that came a little too late, but we took a flask of cells that were not resistant to anything. So they had just been fresh. We divided them freshly, and we treated them with the DMSO, and that's what we dissolved our drug in. And we found that all the cells died. When I treated the cells that I had been making resistant to supposedly TMZ, they survived that high dose of DMSO. So DMSO is already very naturally toxic, which is why I ran that control, was to see if maybe they're surviving the DMSO, not the TMZ. I caught it late, but it was caught, and now we know we're starting with a much higher concentration of TMZ, which means it has a lot less DMSO. And so, I know that at the highest treatment of TMZ that they're getting, they're still going to be able to survive that amount of DMSO.
So your treatments of TMZ are dissolved in DMSO, correct?
Yes.
So how are they administered in humans? Can you replicate that to do it in the mouse cells?
That's like to replicate it in a mouse, you mean, or?
So essentially, you're dissolving. So the DMSO has a really big impact, as you stated earlier, in affecting the viability of these cell lines. And you want to essentially isolate that from the effects of TMZ. So what kind of solvents are they doing in actually administering TMZ?
Oh, in like a human cell.
How do they give those doses in us if DMSO is ridiculously toxic, as you stated before?
Well, the concentrations of TMZ that we use in cell culture are so much higher than in humans. I don't know what they're dissolved in to deliver to patients, but it's about a 200-fold decrease in concentration. So you wouldn't need to use DMSO, I'm assuming, to deliver the drug. Whereas these are 100, 200 times the concentration of chemo that humans are receiving. Did that answer the question?
Yes.
Okay.
Any other questions? Thanks, Shania. So I encourage you to stick around and talk to the presenters. Just another big round of applause for everybody. Great job. And thanks for coming. Look forward to seeing you at the next colloquium next Tuesday.